work with synthetic cells is to understand how living cells even fuse or mate with each other as they continue with their metabolic processes. And currently, we aren't able to mix together very many unique populations of cells together in the same environment without them fusing uncontrollably or without time being a component in that. So to start with, how does fusion, whoops, oh no. How does fusion actually work? So one of the methods of controlling how liposomes can fuse is to embed single-strand DNA into one of these liposomes and a complementary single-strand DNA into a second liposome. And when they are introduced into the environment together, these single-strand DNAs come together and hybridize, and they physically bring each of the liposomes closer together, so within 10 nanometers of distance. And at that point, electrostatic forces are overcome from the buffer system, and slowly the outer leaflet starts to combine, the lipids and cholesterol start to mix, and then the inner leaflet starts to combine, and at that point, the actual contents begin mixing. And this figure down here is just an example of how these single-strand pieces of DNA move forward into hybridization. So in our lab last year, we published a paper, and this figure basically shows one type of complex system where you can use this DNA-mediated fusion. One of these, in one of these liposomes, there is GFP DNA encapsulated. In this other liposome, there is RNA polymerase encapsulated. Each of these liposomes have complementary single-strand DNA embedded in them. And as they are brought together in the same environmental system, they begin to hybridize, and each of the liposomes, now in close proximity, start to actually fuse. When they fuse and their lumen continues to mix, the RNA polymerase comes into contact with the GFP DNA, and GFP is both transcribed and then translated. But the only issue with the system is that as soon as you introduce these two liposomes together, these pieces of DNA will facilitate fusion pretty quickly. So how do we control for reactions before fusion occurs? So me and Kate, we came up with this idea of this 3-part oligofusion, borrowing ideas from the DNA fusion that I just mentioned. So in this, we create liposomes very similarly to how you normally would. So we take phospholipids, cholesterols, we use the ideal buffer conditions, so pH and certain salts, but we embed with single-strand DNA that are not complementary to one another. It's only when we add this third fusion oligomer that the little arms of the fusion oligomer hybridize with those that are embedded in the synthetic cells, and it causes the liposomes to come together and fuse, come together in close proximity and fuse like you normally would. So this allows us to control for the time aspect. It's only when we add the fusion oligomer that, that the fusion between the two liposomes occurs. So the interesting thing about this fusion oligomer is that in the most recent iteration that I'm talking about of this design that I came up with, I have included an I motif sequence into the middle of this fusion oligomer. An I motif is Hoogsteen base pairing as opposed to Watson-Crick base pairing. And in I motifs, a protonated cytosine is going to base pair with another cytosine. And when these I motifs were first discovered, they were happening in highly acidic situations, so anywhere from pH 2 to 5. And these bonds, they happen in such an intercalated fashion that the I motif secondary structures are really quite stable at acidic pHs. And I'll mention in a second the exceptions to that acidic condition rule. But on this figure right here, this is an application example of where DNA has been connecting these two nanoparticle spheres. At first, there is double-stranded DNA, but they include I motif sequences. And in acidic conditions, these I motif sequences form, and they physically reduce the distance between these spherical nanoparticles. But in basic conditions, the I motifs dissolve and the distance between the nanoparticles increases. This concept is especially important for what I'm going to mention. So we discovered this paper by Nestorova and Nestorov, where they rationally designed these I motif sequences to work in biologically relevant pHs, which comes in really handy because all of us, we work with mostly cell-free systems, and those are happening in anywhere of like pH 6 to 7.5. So in this graph, you don't necessarily have to worry about the rest of these curves, but if you look at this bright blue curve, this is what I actually ended up using, and this is telling me that anywhere from pH 6 to pH 6.75, the I motif is 100% folded. So it's in its most contracted form. But as the pH starts to become a little bit more acidic, so very small pH unit moves, but anywhere from pH 6.75 up to pH 7.25, the I motif starts to dissolve and the single strand becomes unstructured and goes into more primary structure. So I used this exact sequence, and I hypothesized that I could put it into the middle of my fusion oligomer, and that would facilitate to bring the liposomes even closer together. And the reason I did this was because the earlier iterations of this fusion oligo, I was doing mostly unstructured DNA, or I was working with loosely structured secondary structured DNA, and it just wasn't working as well. So in this I motif sequence, I put it in the middle between the either of the arms that would hybridize to the embedded DNAs. And in a specific pH, in this case, it was pH 6.65, this I motif would form and would physically force the liposomes together. So after I designed these sequences, I first wanted to test whether they would even behave the way I wanted them to behave, especially since there are arms attached that don't have anything to do with the secondary structure folding. So in this graph, it's just showing that as absorbance at 295, as you decrease the pH, you can see that the absorbance starts to increase, showing that the I motif is forming. So at pH 7.5, the absorbance is quite low, but as you start going more and more acidic, anywhere between 7 and 6.75, the I motif starts to form. And then by 6.65, which is the important pH I'm going to be talking about, this is where the I motif is theoretically 100% folded, or most of the I motifs there are folded. So I took this idea and I tried to do quite a bit of membrane analysis. And then more recently, I started to focus on lumen analysis to see whether the lumen was actually mixing the way I wanted it to. And in the first experiment of that, I took one liposome where I encapsulated a completely independent single-strand DNA that had a fluorescein molecule conjugated to one end of it. And then in another liposome, I conjugated a complementary DNA, but this one had an Iowa black quencher. And the idea was that if they were able to fuse because of my fusion oligomer, the FAM signal, FAM in this case is the type of fluorescein molecule I used, the FAM signal would be quenched by the Iowa black. And what I found, luckily, was that that was true. So in this top diamond signal, I want to see that there is a decrease of the FAM signal over time. And that's what I see starting with at the very top, when I add the fusion oligomer to the system, the fusion oligomer with the I motif in the middle is able to hybridize with both of the embedded sequences and bring the liposomes together. In contrast, this bottom negative control is no fusion oligomer. It's just the two liposomes with their embedded DNAs that are not complementary, so they should not fuse. And the important takeaway here is that this negative control signal is stable, so FAM isn't being quenched to the same rate that FAM is being quenched in my actual time-controlled sample. So after seeing this, I moved on to a slightly more complex reaction. I tried to recreate a little bit of what the other paper in our lab did, where I encapsulated GFP DNA in one liposome, and then in the other liposome, I encapsulated RNA polymerase, but I embedded them with non-complementary DNA. So again, they shouldn't hybridize with each other unless the fusion, the I motif oligo is present. And so I also wanted to see, was my pH actually playing a role in this I motif sequence? And what I found was that yes, it was, because at in this triangle signal, you can see that GFP is being translated, meaning that when I have my fusion oligo present, the liposomes are actually able to fuse, but more importantly, they are able to fuse at a higher rate at pH 6.65, which is up here, than in pH 6.9 or pH 7.5. So you can see that the GFP translation, GFP fluorescence is at a much higher rate. But this is the final piece of data where I wanted to actually see, in contrast to the negative control where the fusion oligo is not present, can I see a GFP translation difference? And luckily, I have been able to see a GFP translation difference where this diamond signal is the fusion oligo present and GFP is being translated at a higher rate than the negative control where the fusion oligo is not present. So to end right there, what we want to do to move forward with this project is, one, think about how we can use this concept in more metabolic development of synthetic cells. So can we perhaps have some kind of transcription translation cascade within one liposome that causes the export of this fusion oligo, and then once that export happens, it causes hybridization and fusion with another liposome and then sets another reaction into motion. And we are starting to do work on there, but I also wanted to mention that this is a technology and I would love to see how this works in other liposome contexts. So if any of you have any ideas and you'd like to collaborate with us, please feel free to reach out. So with that, I will say thank you to all of you for listening, but also especially to my lab. It's one of the most supportive work environments, and I'm really grateful of that as a grad student. And of course, without Kate, I wouldn't have actually started working on this project. This was one of the first projects when I joined the lab. And I'd also like to say thank you to Aaron, who steered me towards this eye motif paper. And the fact that half of our lab works on G quadruplexes and eye motifs has been like a huge boon to its collaboration with my work. And of course, Tanner is a grad student as well in our lab, who's really walked me through a lot of the oligomer analysis using UV vis and circular dichroism. So I'll end there. Any questions?
Great. Thank you very much, Judee, for a very nice talk, and nice to hear about your research. We do have some questions. I will go ahead and just read these out. Tom R. Is asking, why does the negative control not start at the same initial level as the experiment?
Sure, yeah. So I think one of the reasons is that there is some autofluorescence with the TXTL that's encapsulated. The other reason is that I haven't yet controlled for any of the extra components that are not encapsulated. So it's possible that in the environment, there is some TXTL components like RNA polymerase or even DNA that's coming into contact with each other and producing translated GFP, but that is not encapsulated. So actually the next thing that I'm going to be working on right after this experiment is adding some proteinase of some kind to the external environment to get rid of that and to also control for autofluorescence.
Great. Felipe Quiroz asked, I usually imagine the need for having some liposomes at high density. In that context, DNA-triggered fusion is a clever approach to introduce a logic to fusion partners, but have you, through the use of your approach, have you, though, thought of using your approach to control the reverse unintended fusion at high density liposomes?
Can you potentially explain this question a little bit more? Have I thought about using my approach for controlling reverse? Yeah, I mean, I wonder if my interpretation of this question would be, if I have a highly dense population, would this approach work in preventing unintended fusion? And the interesting thing is, I have actually tested higher population densities of this system, and at least with the older iterations of this fusion design. And I will say with the older iterations, higher population densities really starts to negate fusion control. I have yet to test this with the iMotif sequence, but even if you double the current population density of the data I showed you, you start having a lot of unintended fusion. So I think that's actually one of the problems the field might have to solve in the future. Like, how do we balance the population density with how much translation or even transcription the liposomes are capable of doing? I hope I interpreted that question okay.
I think that was reasonable. Marcos is asking, have you tried these experiments in GUVs to directly observe the fusion events?
So these liposomes, or at least the data I most recently showed you, the liposomes are anywhere on an average are about 10 microns across. So they are quite big, and this is all like fluorescent indirect measurement. So one of the things that we want to do in the future, especially before we publish, is do a lot of microscopy work too. So maybe that would be another direct way of observing fusion. I am currently starting to work with 30 micron liposomes as well. So hopefully that covers some of the GUV ideas.
Yeah, I think Marcos is just interested in whether you were intending to do confocal or other, microscopic observation, and it sounds like you are going in that direction.
Yes, definitely.
Tom Arts is asking, have you also quantified the fusion efficiency? What percentage of vesicles do successfully fuse?
Yeah, Kate and I were just talking about this, and we are working on how to quantify fusion efficiency. I mean, one way would be to put these liposomes under a microscope and count how many are fusing over a period of time within that field of view. But that is quite hard, and I think we are thinking about new ways, but also if you have any good suggestions, any of you have any good suggestions for what we can do to better quantify efficiency, I am definitely open to hearing that.
Thank you. I hope I get this right. Misko Maika is asking, have you considered producing a system that would release the contents of a smaller GUV enclosed in a bigger GUV into that GUV in a time-controlled fashion, a situation analogous to lysine cell nucleus inside the cell?
No, that's a really interesting thought. I haven't really thought about that specifically, and I wonder how we could set up this DNA-mediated scheme that we could have this smaller GUV release inside. Yeah, that's a really interesting thought. Thanks for proposing that.
Okay, let's see here how we're doing on time. We have time for maybe one or two more questions. Yi Lai asks, is this possibly turned into a DNA sensor / diagnostic device where DNA or RNA can trigger this response?
Yes, definitely. I mean, that is also one of the things that we would love to consider as an application. Yes, that would be very interesting, especially like if we could test it with living cells and see how that works, then that would be really cool.
A few people have asked about the lipid composition of your experiments.
Yes, so originally I was working with POPC, POPE, and cholesterol, and then eventually after reading the Chan paper where they showed a lot of this single like normal complementation work, I switched to DOPE, DOPC, and cholesterol, and the concentrations I'm currently working with are about 1 millimolar of each DOPE and DOPC.
Okay, final question. I think it's related to the earlier question. Could you control the fusion by small molecules? For example, if there was a motif inducing ligand, could it induce fusion?
Motif-inducing ligand that induces fusion. So yes, you can control fusion with small molecules, first of all. There were some people early on when people were looking at liposome fusion, this whole idea of bringing together liposomes was from the snare-mediated proteins. And if you use that kind of idea and you think about other proteins that also form partners or are homodimers and they come together, you could perhaps use some kind of small molecule ligand to bring those two protein parts together. And that's actually a different project that is kind of tying into this whole inducible fusion as well that we're working on.
I see. Yeah, I think there's been a lot of work in the DNA molecular logic where you could in principle incorporate an aptamer that would bind some arbitrary small molecule that could lead to change in nucleic acid conformation that would ultimately mediate the fusion. Okay, well, thank you very much, Judee, for a very nice talk, and thank you for everyone for asking the questions. I apologize, we had lots of questions. We weren't able to get to all of them, both for Judee and for Michael Jewett. I would encourage people to email Judee and Michael if you have burning questions that you would like them to ask. At this time, I would like to just mention what we have coming next week. As I said at the beginning, this is a weekly seminar series for the next 10 weeks. And so at the same time, next Monday, 10 a.m. Mountain, our speakers will be Ramin Golestanian from Max Planck Institute for Dynamics and Self-Organization. He'll be talking on how living matter self-organizes while breaking action-reaction symmetry. And Jan Steinkuhler will be the second speaker from Max Planck Institute for Colloids and Interfaces on controlled division of cell-sized vesicles by low densities of membrane-bound proteins. And finally, I would just like to thank sponsors for this series. Primary sponsors, the National Science Foundation. Other sponsors include University of New Mexico, the Max Planck School Matter to Life, Build a Cell Network, the New Mexico Consortium, and Los Alamos National Laboratory. Once again, I thank you all very much for joining today, and I hope that you will continue to join in the coming weeks and our conference coming in May. We will send out reminders of this information in the coming week. Thank you all.